Okay, I think we should start. So our next speaker is Talia. That's about the shape of things.
Right. So my work is mainly focused on the problem of high-level vision. So you look out, you see a detailed world, but actually this is just a reconstruction from patterns of light hitting your eyes, the back of your eyes, and making their way through your brain. So how does the brain turn something like this into something meaningful? And the story starts with a hierarchy of visual areas that go. There it is. Yeah, so information gets relayed through the thalamus and gets processed in V1, V2, V3, various stages along the ventral stream. And you can see that same motif echoed in recent advances in computer vision algorithms, these deep learning ones that you've heard a lot about, where we have successive layers or representational stages before you get to some output. And in much of the work to date, along each of these stages, we've been trying to figure out, well, what are the stages and what features are represented? So we know that there's relatively low-level feature information in early stages, mid-level feature information somewhere in between, en route to higher-level representations that can give you the categories of things. And in trying to understand these, the current focus is usually on like layers and areas and hierarchies. How is this information transformed as you go from input to later stages? But what I want to argue today is that there's value in thinking about an orthogonal view and what runs across the hierarchy. And the reason why I think this is important is because biological brains have topography. Tuning problems aren't randomly distributed. A neuron here and a neuron next to it can have similar tuning. You can see this, the clustering of phase-selective units in the fusiform gyrus. You can see this in large-scale retinopathy across early visual cortex. And these are usually linked to the idea that there are wiring limits. You can't connect every neuron to any other neuron you want because there's limited space in the brain. So you got to organize it efficiently so that the things that are nearby don't take as much wiring to get to them. So in this sense, where you put things matters. So you might be able to infer function from the topography. And it's generally true that the spatial organization of our sensory regions are highly preserved across mammals. And topography is relatively absent from TNS. It's not a constraint put on there. But today I'm going to try and argue that topography is critical to think about information readout. So how you get from the visual system out to the rest of it. So today, there will be two parts, hopefully. One is about the organization of object responses and this orthogonal view. And the second is relating brain measurements and behavioral measurements, and the speculative story about maps and readout. And just as to get you oriented, these are largely empirical findings, but where the idea of geometry comes out is in trying to make sense of how they go together. So at the very end, you'll get to see how we're thinking about this as a high-dimensional space with the geometry. But it's gonna be mostly empirical throughout the beginning. Okay. So to get you oriented, this is an image of ventral stream responses to objects in the human brain. So it's partially inflated. This is the lateral view. This is standing sideways looking this way. So if you rotate the brain underneath, that's looking at the underneath side here. And this is if you put someone on the scanner, you show them pictures of objects versus just looking at a fixation cross, and you drive responses largely in the back of the brain. So this is the ventral stream. The question is, how is this content organized? And for a while, our model was this Raisins-in-Oatmeal model. So we knew that there were some regions of this cortex that showed selectivity to certain kinds of categories, faces, bodies, scenes. But there was no such region for cars and shoes. And so, but we did know that if you measured a response across all of this cortex and look at the patterns to cars and shoes, you could tell them apart. So the idea was that there are these sort of large-scale clustered regions for some categories, and then everything else was in this finer-grained structure that was beyond the limits of fMRI to measure. But in thinking about what objects have, if you just think about all the objects, one thing that's true about them is that they all have a real world size. Some things are small and you can hold them in your hand. They tend to be curved because we make them for our hands. Some things are big. They have larger surfaces because they have to withstand gravity. And we navigate around them and they have different affordances. So there's a property that holds across all objects that has a lot of visual relevance and behavioral relevance. And so we tested the idea that there would be a neural division of labor, but some cortex might be involved in processing small objects more than large, and vice versa. And in fact, that's what we found. So when we scanned people looking at pictures of big objects versus small objects, these are all presented at the same visual size on the screen. So what's differing is only that they are big or small in the world. And you can see across this cortex, there's this large-scale organization. So blue, this cortex responds more to big objects than small to big, then small, then big. So it kind of is mirrored on the lateral ventral surface. And another distinction that's had been noted before was the distinction between animate and inanimate entities. So this is primary division. You can see early biases and sensitivity to these distinctions in young babies. You can see neuropsychological patients that have deficits recognizing one kind, not the others. There's lots of reasons to believe this would be an important division. And indeed, if you look at this organization across the brain, so showing pictures of animals versus objects, you can see this cortex prefers objects, animals, objects.
Yeah. In the previous,
it's coming.
For the food.
Oh, wait, say that again?
Like in the previous slide, what would food have a different character?
Would food? We haven't scanned food specifically. There are bits of food items. Usually they're small. So we did, food was, there's like the bow tie pasta, but I haven't scanned like food as a class separate from inanimate things, so that's a good question. Yes?
Do you have paradoxical pictures where there's a small object physically, then you put it next to a big object such that it should be interpreted as bigger than what it should be?
Yeah.
And how would that just move in the
What we did was instead of, we did it in mental imagery. So we had people imagine apples and pianos, and then we had them imagine giant apples and tiny pianos. It was awesome, in fact. And what if this region were really about an abstract sense of size, the size you know the thing to be, then you should flip the organization, but you don't. It stays exactly the same. The regions that like apples likes apples and giant apples. The region that likes pianos like big pianos and tiny pianos. It's not about the size in an abstract conceptual sense. It's something that's related to the size that doesn't change when you think of it as big or small. Probably the shape, which we will get to in a second. So yeah, that's good. Okay, so for the animacy organization, this had been mapped before, but had mostly been focused on, like just down here, and we just mapped the broader organization. You can see that it alternates on the lateral and ventral surface too. So the next question we asked was, how do these dimensions go together? If you think about it, we have this nice two-dimensional space. You can divide things into animals and objects. You can separately divide those into big and small. So if we know the size organization is like this, then you should map your animacy organization like this and preserve the 2D topography right on the cortex. And this isn't what we found. After a long time analyzing, we figured out that there's a tripartite organization. What that means is there's cortex that likes big objects more than everything else. All animals, but didn't care about their size. Then small objects. So the size dimension only applied to the inanimate objects. The cortex that liked animals, liked all animals. If you try and contrast big animals versus small animals, it's too fine-grained to see. You don't get any systematic organization. This large-scale tripartite organization also helped make sense of the category mosaic. So these often looked a bit hodgepodge. Why are they where they are? But if you see this, the scene regions sort of fall in the zone of cortex that like big objects more than small objects. And this is consistent with some of the stuff Ed talked about. The face and body regions are not unrelated to what's around them, but they're meaningfully related to the responses to animals. One second. And yeah, and the small objects there. Yes, yes.
So I'm wondering how we should think about the relative size of those areas on the surface of cortex. I mean, does the number of stimuli you use help you make these areas bigger or smaller? Do you think this is, this is something, there's something true about this and we need to think about?
Yeah, it's a good question. One thing that makes me think it's not about the number of images per se, I think you have to get it up to sufficiently high. Like if you did 4 animals, like you're not sampling the space of animacy as well. But if I were to like inflate this slightly less, you'd see that these follow the major soul sign gyri. So actually, like the large scale functional divisions kind of, you can know where they are just from the anatomy by looking at the folds, so that they're not too far off from the folds, which makes me think they probably wouldn't change too much even if you had many, many more images. Yes?
There seems to be like a separate location between like small object and big object. So in terms of size, when do we transition in size? Like let's say something in area like yellow versus area blue. What is the size basically that segregates something from being small to something that
Oh,
is
right, like why that's the continuum.
What's the
And
size?
what the heck is the animals doing in between there, and what's the break? Yes, that is a very good question. That one, so we did an experiment which never got published in the end because the reviewers made us take it out, but that's another story, where we varied the size and looked at the continuum of responses. So it turns out in all, oh geez, sorry. Oh, I'm not used to this clicker, it's my fault. There it is. The pointer is sneakily close to the other buttons. Okay, so in this cortex, it's not like it doesn't respond to small objects at all. It's actually parametric. So it responds most to big objects and then pretty systematically down to small objects. And the same thing is true here. It's relatively parametric. Though, if you had to draw a distinction, like where that cutoff is, it's like chair-sized and bigger ends up here. So it's like versus, hand size and smaller. And these intermediate sized objects, there are actually not too many of them. They're like coolers and desktop fans, like things that are like 2-hand but kind of stable. Those I haven't like systematically explored, but for the most part you end up with like furniture and chairs and buildings drive this the most, and less so tools versus the other way around. Does that answer your question?
Yeah, I'm sure you haven't done cross-cultural studies, but I'm wondering where expertise affects someone who knows lots of animals might have a discrimination between
Lower organization, but
small and
it would be
large
interesting
animals.
to see that actually, because it's true, like we learn all our animals from pictures and books, like it's not like we have a lot of visual experience with animals. So, one interesting idea is that what we do see a lot of people, and it's like
Yes,
possible
yes.
we're understanding animals by using our models of human
That's
bodies,
right,
but that's another project. Yeah, cool, okay, so there's this meaningful organization encompassing this category mosaic, so we've kind of linked the other objects to the category-specific areas. And the question that I think raised was like, are they really about size? Really? Are they really about animacy? Really? And we did that experiment where we made people imagine them the wrong size to know it's not size in that very abstract way. And generally, this raises a challenge for interpreting neural responses to real-world objects, which is whether it's neurophysiology or fMRI. You get some measurement in some region where big objects represent more than small objects, and you don't really know why. Is it something about the abstract sense of size, or is it something about systematic differences in their shape or their texture, or even higher level systematic differences in where you find these in the world or what they're used for? And maybe an interesting idea is that you get a different answer all along the continuum. So early, regions here might be reflecting these differences, and later regions here might be reflecting these differences, and there's probably somewhere in the middle that's reflecting mid-level differences. And so, but because it's so hard to tell these apart in real-world objects, is that there's actually a pretty massive debate in our field where a lot of people think all of this can be explained by low-level differences. Some people think, well, some of it's probably mid-level. Others think the whole thing can be explained by high-level differences. And, presumably some of it's mid-level. Many people think that, but we haven't had a good model of mid-level vision to really get at this difference. So how do these various levels map along the hierarchy? And to get at this question, we created a novel stimulus task called texforms. And I'm just going to introduce you to them by also having you simulate what it was like to be an fMRI experiment at the time. So, by the way, these were generated by Bria Long, who did all of this work. She would be on the job market very soon. I can hire her. She's fabulous. So here's a demo of the fMRI protocol. I would like you to clap when you see an image repeat back to back. Everyone ready? Also keep your head very still. I got my joke. Excellent. All right. Good, very good. You're awake. Good. Any idea what you saw? You didn't recognize
It was
it?
maybe a squirrel.
Okay, here is what you saw in the exact same order. We didn't tell participants this, but they actually saw these image
No.
designs.
Really?
No. So, oh, you stopped paying attention and doing it. Yeah, we're all falling asleep. Oh, there's the second clapping. All right, so these are text forms. We generated them by running original images through this texture synthesis algorithm that was modified by Freeman and Simarity. And you can see that they look like blobs, but they look like different blobs. There's systematic structure that's been captured in the differences in texture and coarse form information, hence the name text forms. And these, of course, if you haven't figured it out yet, correspond to big animals, big objects, small animals, and small objects. So now we can ask, how do text forms drive the ventral stream? Where our predictions were, if you look at the responses to original recognizable objects, we see these sort of elongated spokes along each domain. So it made sense that, like, well, maybe if we're thinking about, these are little mini hierarchies. And so in the posterior axis, you see the low level, and maybe what we'll see with text forms is we'll only drive the mid-level regions, but we won't drive any of this cortex that's processing higher level aspects of the stimulus that would require recognition, or even just any sort of sense of actual 3D geometry. You can see these text forms are pretty primitive. So we were expecting to kind of isolate this mid-level of representation, also not what we found. So let me show you. This is the response to original recognizable images. This is in just one participant. So looking at their big object preferences, animal preference, and small object preference. So it's like a three-way winner map amongst these. And then we can do the same thing to see what happens with the text forms. So effectively, we get the exact same thing. Very similar patterns of activation. They're weaker overall, but not too much weaker, despite the fact that they have very different high-level content. So our idea that we would isolate a more posterior aspect of this hierarchy went out the window, and instead now we've started to speculate that maybe this entire bank of cortex is a massive mid-level feature bank for recognizing the shapes of things. And we pulled some insights from deep nets to get at a sense of what the level of representation is. And so this is drawn from what you saw in the earlier days. So you can take a deep model like AlexNet, trained to do 1,000-way object classification, and then you fix all those weights, but now you show it text forms. It thinks all the text forms are plastic bags. Nonetheless, you can still measure responses to each of these text forms in every neuron, and then try and see how well do those deep net neurons do at predicting brain responses. One level of detail, this is not important to understand per se, but the way we do this is by trying to predict the representational geometry. There's a problem with how do you make a box, how do you do a correspondence between the brains and the nets, and the way that field is typically doing that. So sneakily, I showed you 4 conditions, but actually there were 24 conditions. And so what we can do is look in these voxels and build a 24 by 24 matrix where what we have is how similar the pattern of activation is across all of these voxels. And so things that are very similar are in dark blue, and things that are more dissimilar are in yellow. And this, for what it's worth, is called a representational geometry. That is, it's basically, you can think of it as the set of all pairwise distances between these 24 positions. We can make this is nice because it's dimensionless, so it's 24 by 24. We can do the same procedure for each layer of the deep net, and now look at their correspondence. So it's a little bit more complicated than that, the fitting, but for all intents and purposes, it's basically that. And so when we look at the data where we look at how well deep net responses to text forms predict neural responses to text forms, that's on the y-axis. You can see that by about layer 4, and then maintained through all the high-level layers, there's a pretty good prediction. This is in the context of the noise ceiling of the brain data, which is how well any one brain person subject predicts the rest of the subjects. So if our model can get into this gray zone, that's good news. So basically, this is telling us that it's these mid to late layers, these features of intermediate complexity that are explaining the responses. We can also look to see how well responses in the deep net to original recognizable images predict brain responses to original recognizable images. And again, we do actually pretty well. So this is known. This was one of the things that launched the cognitive neuroscience deep net revolution, which is that, wow, this is the best model of predicting brain responses to date. One thing that's maybe not emphasized as much is look how many layers do very well. So it's not the case that, different layers correspond to different parts of the hierarchy. And when you break it out that way, you can kind of see that result, but the dominant result is that all of them do pretty well across much of this cortex. And then you could do something interesting. You could take the deep net responses to text forms and try and see how well that explains the responses to original recognizable images. And so those results are here. So if text forms captured everything that original recognizable images have, you might imagine that the bar would be here. So basically this little gap tells you how much we're missing. So most of the responses to text forms, most of the structure in the brain responses is captured by the text forms. So, yes?
Can you just repeat what actually the prediction task is? So you show it these 24 things, and
So you construct okay, here's the procedure. So let's take 1,000 voxels in the brain to 24 images. We have 1,000 by 24 matrix. You core coef that to make a 24 by 24 matrix. So basically each cell has how correlated the activations are. And then you can do that same thing for neurons in the deep net. Let's say there's 6,000 neurons in layer 2. We can measure their response to the 24 conditions. We can make, again, a 24 by 24 matrix that lets us know how similar are these conditions to that layer. And then we can compare those two by correlation. Does that make sense? I feel like I should draw it. I didn't work through the whole pipeline.
I'll ask later.
Okay. Okay, so to summarize part 1, I told you first that there's this tripartite large-scale organization by animacy and size, but then I also told you that there's mid-level features are basically explaining most of the response structure. So how can this be? Is this cortex reflecting high-level object categories and distinctions, or is it reflecting mid-level shape? And what happened to the hierarchy? Yes?
Just for me to keep
Yeah.
So when you're doing the text form versus the real object, are the same other brain region active? Like because you're imaging the whole brain. So with text form and with real objects, other brain images are still active in the same way. I'm just thinking of cortical, feedback or cortical connection from other areas.
Right, like how much could feedback be playing a role in this? It's a good question. One thing that's interesting is that the deep nets in this case don't have feedback. They're all feed-forward models. So to the degree that they do pretty well fitting the brain responses, it suggests that you don't need to invoke feedback mechanisms to...
They didn't do well on the text form, right?
It did pretty good, but it's true, there's room. So maybe text form, and we do have evidence that even though you don't know what those text forms are, you can guess to some degree whether it's an animal or an object. And so likely that information is getting all the way through, even though it's not for sure that's an animal, there's some probabilistic sense that you know what it is, that could feed back and help amplify differences. Maybe some of the response differences we're not accounting for could be explained by that mechanism. But an alternative is that there's just also a lot of visual features that are different and missing from those text forms, like they don't have edges or contours. So I actually think it's more, the gap between originals and text forms is probably related more to like visual features than extra feedback mechanisms. Don't know if that totally answered your question, but okay.
Would it be that maybe because fMRI signal moves across a large population of neurons, then individual neurons are sensitive to objects, but what fMRI mostly sees is kind of the average of these neuronal responses that are selective, but they come out as mid-level features after averaging.
I mean, I feel like showing the text forms themselves, where we've stripped out a lot of that high level content, and still seeing these activations, argues against a strong form of that account, where like this is an artifact of averaging, because this is just for text forms drive, like we might not expect to see anything in that case, which is what I actually thought would happen. It's like we just wouldn't have enough featural content to drive those areas at all, but then we did, so then we had to make a story for why. Yes.
I know the text forms take away the recognizability of it, but do you have some kind of formal characterization of what image properties are preserved and removed in the text form?
Yeah, so we've been working on that question a lot in a number of different ways. So they're made from the Freeman and Simarity model, which is picking up on cross and the class of features that matter the most. You can make knockout text forms where they're missing different classes of features. The ones that matter the most are cross-scale, cross-orientation ones. Which what that means is you have to be picking up on the way orientation changes over spatial scale, which is kind of like curvature. So then we also got perceptual ratings of curvature. You put a text form on the screen and you ask people, how curvy is this on a scale from very curvy to very boxy? And actually that one-dimensional curvature rating explains like 50% of the structure. So there's something about curvature that seems really important in explaining these responses. Beyond that, we just know like DeepNet level 4, so they're intermediate complexity, low-level visual models don't do a good job explaining this. There's something about continuous contours over a medium scale, that's the best I got. Okay, oh goodness, 1 and then 2, yes.
So I'd like to add a comment to that, because we've been thinking a lot about what that changes, and
Yeah.
what it definitely destroys is large continuous contours. So it's like taking an object image and converting it to a texture patch. So it maintains two-point correlations that are repeated through the image, but not two-point correlations that are not. And so any contours that are there in just one part of the image would be completely.
So this is one thing where the algorithm we used is one step beyond the Portilla and Simarity one, because we did texture in local pooling regions. So it's like we get a measure of the texture here, and a different measure of the texture here, and a different measure of the texture here, and so that way you don't, because otherwise it just fills the whole screen. So this is like localized texture measurements.
Yeah, I was going to ask you what you meant by, modify.
That's right. So where we modified it is we kept more of the spatial structure than is typical in texture, pure proper texture algorithms, which just give you something that can be tiled over a whole screen. Yeah.
I was just wondering, a rich natural environment would be more curvy, not just.
It's a good question. I know, I spent a lot, I think of like built man-made artifacts where you have all these edges, but then in the natural world, the statistics of the natural world are different than the statistics of the manufactured world, and how that affects this, I do not know. I think that's a really interesting question, like how much of these are preserved, because there's enough reality to the fact that large surfaces tend to have long lines, and things that are tall withstand gravity, they tend to have these edges. So I think that's a really interesting question though. I genuinely don't know. I could see it going either way. Yeah. Okay, I have 15 minutes. This is gonna be interesting. Okay. I'm still gonna go through this because I think it's an important side theoretical point, which is trying to make sense of both those data at the same time raises a question about what your theory is about what neural regions do. And one classical side of theory says that neural regions isolate levels. So different regions should do different things. They isolate levels of representation. And on this framework, we do a lot of experiments where we try to control for one level to isolate another level. So we'll control for shape to isolate category. And that's sneakily trying to assume a model underneath where there's maybe some cortex here that's, say, about shape, that's different from cortex here that's about something high level. And it assumes that it's possible to break those links. But I actually think, if you think through this pretty deeply, there might be a deep flaw in the logic there. But a way to rescue it is to think of this alternative way of thinking about what neural regions are trying to do, which is to capture covariance. And shape and category are intrinsically correlated in a meaningful way. And so in this sense, mid-level features are related to the high-level categories. Maybe big things tend to be boxy. Oh, see again with the clicker. Big things tend to be boxier. Small things tend to be rounder. You can measure this from just image statistics. Animate things maybe tend to have leggier things. We're still working on that one. And it's worse. It's not just shape and category. Big things tend to be experienced in the periphery and have more high spatial frequency. Small things tend to be experienced in the fovea. When they get out in the periphery, they're beyond your acuity limits, so you only really see them and recognize them when they're in your fovea in the center visual field, and they have more lower spatial frequency. Big things are also important for non-visual things like orienting and navigating, small things socializing or grasping, animate things socializing, and so all of these are correlated in a meaningful way. And in fact, in the region of the parahippocampal place area, there's independent evidence that every single one of these drives that cortex to some degree. It's simultaneously driven by objects that are more navigationally relevant than not, when they're those that are at boundaries versus not, and by just presenting checkerboards in the periphery relative to the fovea. So this is a bad thing if you're trying to isolate the level of what the brain region is doing. But I actually think this covariance in the world between low and high level features is a great thing for brains. It's exactly how we understand how you get from patterns of light into meaningful and useful representations of objects in the world that are useful for behavior. So this is the orthogonal approach set out one more time, which is if we're zoomed in looking at the ventral surface, we've spent a lot of time thinking about hierarchical processing. But I think the data show it's a blurry gradient at best, and there's a lot of it that's mid-level. But what's clear as day are the content divisions that run through them. So you can see this cortex care more about big objects, animals, small objects, so these are grouped by high-level category, even though they're reflecting mid-level features. So why would that be the case? And there's two different theories that I think are not at all mutually exclusive. I think they're mutually reinforcing. Which is, one is that there's just systematic biases in the input. So if you think about a person walking through the world, you're looking down at small objects. People tend to appear at a certain visual size. Large objects tend to have a more visual angle. And so in the cortex, just adjacent to object cortex is early visual cortex that is organized by fovea and periphery. So it might naturally be the case that small objects experienced in the center visual field end up sort of aggregating and recruiting and end up landing there, whereas big objects experienced in the fovea, periphery end up sort of aggregating there. So we have this sort of just natural progression from just extract more and more covariance in interesting ways. At the same time, it may be the case that these particular divisions are useful, and there's lots of possible ones that could have come up, but these ones are reinforced by long-range networks. So these are the kinds of white matter tracks that are set up way ahead of time. You don't grow new white matter tracks. You prune them away, but they're largely there. So we know that this region connects mostly to like retrosplenial and hippocampus, things that are involved in navigation. This region here has strong connectivity up into spots in the parietal lobe for reaching. This region here connects to the social brain network, medial PFC, STS. So it's possible that those long-range connections help enforce these particular content divisions, even though what they're representing is the shape of things. So on this view, of the visual system, I think maybe trying to isolate the level of representations along the hierarchy may actually be impossible, but trying to understand and characterize these content channels that run the other way may be a productive way forward. There's 10 minutes left. So one option is I can take questions here and just end. The other option is I can breeze through this and get to the end. I will take a poll, take a moment to consider. Raise your hand if you just want to ask questions. Raise your hand if you want more data. More data, right. Okay. Brain paper correlations and a speculative story about maps and readout, and I'm going to fly through this. Here we go. Also, this is the work of Peter Cohen, who is a professor at Amherst. Okay, so visual behaviors have some constraints. We can't attend to everything at once. We can't instantly find what we're looking for, even if it's in front of us. So here's a little demo. Try and find this object in this array. Sneakily, there's 3 of them. Keep looking, keep looking. There's the one you probably found first. There's the one you may have found second. Here's the one you probably found when I told you to keep looking for something. So intrinsically, how fast it is to find a thing depends on the other things around it, what you're looking for. So it's related to the targets and the distractors. And there's lots of work trying to identify what properties and features of these objects help you find something faster or slower. And among the probable guiding attributes are things like shape and curvature. And largely, if you allow me a gloss, this list is mostly about the way things look rather than about what things are. And so what we did here in this project is use visual search speed as a behavioral index of visual similarity. So here what we did is we measured a behaviorally relevant similarity space among object categories using visual search. Then we measured brain responses to each of those categories. And then we tried to see where does the similarity relationship we find in behavior emerge along the visual hierarchy. So the stimuli used were 8 different categories. They span the animacy and size continuum, so there's a trade-off in how many you can use, but we tried to pick a big joint. We had 30 exemplars per each of these with lots of different variation. You can also see they're hard to see. That's because they're matched on lots of low-level things like global spatial frequency and luminance and contrast. So here's the demo of the experiment. I want you to shout out, is there a face? Yes or no?
Yes.
Good.
No.
Harder, right?
Yes.
Yes.
So that's what they did over and over and over again. Sneakily, the objects that were the distractors all came from one of the other categories. So it was a face among cats, a face among chairs, a face among cars, a face among shoes. And then we can measure reaction time to find a category amongst the other categories. And we tested all target distractor pairs. So faces among hammers are the fastest with reaction times around 850 milliseconds. Cars and phones were the slowest with 1.5 seconds. So this is a huge, actually 500 milliseconds, a pretty big behavioral range, where the theory is that then that means that these are more different in some feature space that we don't know. But it's easy to find a face among hammers, whereas cars and phones, they're more similar because it takes longer to find them. Making sense so far? Good. So then we gathered neural measures to these 8 categories. I'm going to skip through these details. The key things are they're in a new group of participants, and they're measured one at a time. So any match that we see between brain and behavioral measures relies on a pretty stable representational architecture that doesn't change based on the task you're doing or even your own brain. Just to generalize across brains. Okay, and we did a representational similarity analysis. So basically for all pairs, we have the search reaction times, the time it takes to find one amongst the other. And then we can do the same thing with the neural similarity. So we can take a region and we can look how correlated are the responses across between the categories in that region. And we can just look to see how similar are those. And the trick is now to vary the neural region and try and figure out where this behavioral similarity emerges. So the first thing we did was these large-scale sectors. So early visual cortex, ventral stream, we divided into the lateral and ventral component and dorsal stream. Long story short, basically in all the high-level visual regions, we see a really strong correlation between brain similarity and reaction time. So here each dot is a pair. So that's like faces among, cars among, more similar is harder. So yeah, cars and phones might be here, and all the face-offs might be there. Basically, how similar the patterns evoked in these regions very strongly predicted how fast you could find one amongst the other in visual search. And that was true in the ventral, in the lateral, occipital-parietal even. The only place it wasn't true was early visual cortex. And that was in effect by design. You'll notice all the neural similarities are really close together. We did all those different views of faces and stuff to make sure they didn't have really similar anatomic footprint. So we kind of ensured this was the case. Okay, so we basically see moderate, strong correlations across the dorsal and ventral screen. Next, we did a searchlight. And what that means is we, for instance, we know that in this region there's like face regions and body regions. Maybe it requires pooling over all that category mosaic in order to get this structure. So you can take a smaller region and just look everywhere and look to see if you can still see the correlations there. Also lets you look beyond the visual system. What we found, is that basically everywhere you see red there's a moderate correlation with the structure of the neural responses and that visual search structure. So basically the entire visual system, even at this mesoscale, predicted behavior or related to behavior. Prediction was associated with behavior. And that's surprising because we know that my computer is going to run out of batteries in 5 minutes. No, let's just go.
Do you need a part?
I'm trying to trade off the 3 minutes that is likely to, you know what, I'm just going to go and it's computerized the text. Okay, so we can look in, say, FFA, a region that we know cares more about faces than other categories. And we still see that this relationship holds, even in face-selective FFA. And it's crazier than that. When you take out all the pairs that involved a face, so now you're just looking at the similarity among, hammers and shoes and things, you still see the same relationship holding. And you see that not only in FFA, but in PPA with the buildings removed, and in EPA with the bodies removed. So even in category selectings, with selected regions, without the preferred categories, this relationship held. So we ended the paper there because the implications of that are a little crazy and we actually couldn't get it published. So I will summarize where the paper ended off, and then with my 2 minutes, I will speculate on what I think it might mean. So we measured this behaviorally relevant visual search space, and we basically found a strong behavioral brain match everywhere. We did a bunch of other analyses that I'm going to skip, and we did a bunch of other tasks, not just visual search, but working memory, masking, categorization. Basically, anytime you're measuring similarity and competition between categories, it's related to the structure in this entire cortex. Which we think means there's a stable architecture of object recognition that's a primary bottleneck for many visual behaviors, and it's related to this shape of things, this mid-level feature similarity. Okay, so now we'll speculate a little bit wildly. So I'm going to roll back to that last result to try and understand what that means. Now we're going to get into the geometry a bit. So how can it be? Most people think of face regions as having face-specific tuning, right? Detect 2 eyes, a nose, and mouth. But why would those kinds of features meaningfully predict the relationship between chairs and hammers and shoes? So if you think about this for 6 to 8 months, you end up somewhere with this conclusion, which is the tuning must not be for face-specific features. Whatever the features are, they have to meaningfully apply to all categories. And we pushed it a little bit further. It's not just the case. So what you're looking at here is all the different regions in high-level visual cortex and how correlated the geometries are with each other. So FFA and PPA have really similar relationships. So there's basically the same representational geometry in each of these regions. And I think that's actually pretty consistent with this finding we're seeing, which is like, this is this extensive mid-level feature thing. And so there's a difference between univariate preferences and multivariate preferences. FFA and PPA, they like different things. They like spaces, less so buildings. PPA is practically reversed. But if you look at the pairwise similarity amongst these categories, they're very highly related. Same pairwise distance relationships. So the preferences, the univariate responses, are telling us something different than the patterns. And I think what pattern similarity is telling us is that they have a common representational goal, which is these features should be tuned to distinguish all categories from all other categories. That way they're not face selective per se, they're just trying to separate all the different things from everything else. And it turns out faces live over here in the part of the space, and I think big objects live over here because they're pretty different in the shape of things. So, and I think deep nets really support this idea, because what are the features learned in deep nets but just trying to understand how to pick up on image statistics to tell 1,000 categories from each other all at the same time. So if this is the case, FFA and PPA are doing the same thing. They're part of the same feature thing. But why have them both? And this is where readout comes in. I think you can, if you just make the assumption that the brain needs high firing rates for readout, which may or may not be true, but let's just go with it, then faces are high on some features and low on others, and scenes are high on some features and low on others. And so you really need to rotate the shape space around so that different parts of it are encoded with high firing rates. And that way, each region can signal different kinds of content with high firing rates. I think that makes sense of some of the puzzles in the literature, which I'm not going to go over because we are 2 minutes over time, and this is the last slide. So I think putting it all together, we have this high-dimensional shape space, which is common. It's trying to distinguish all things from everything else. And it's mapped onto the cortex, where each region is elaborating different parts of this space. So in that way, it's mid-level. But it's doing that so that the wires can come in and read out the right part of the space. It doesn't have to connect to everything. It can just listen to the right part to enable system-level readout. And I think this is where you go through like a mid to high-level conversion, and animacy and size are probably related to the major axes through shape space. And that is the end. Thank you very much. Computer, maybe. I think there are not really time for any questions. Okay, I'll take one and then we'll pass it on. Yes.
So I'm a fan of the inherited retinotopy idea, but
Yeah.
another implication is that the most ventral area should be inheriting the upper periphery,
Yes.
and the most dorsal should be inheriting the lower. Do you have any
Yeah, so
thoughts
we've
about?
looked at that. So yeah, so the fact that it's mirrored makes sense because these bands jump around, but these ones correspond to the lower visual field, which go up to the parietal lobe, and these ones correspond to the upper visual field, which go down to the hippocampus. When you look for upper and lower visual field biases, they're very minor. They're like hardly there at all, which is surprising. I don't know what to make of it. It should be there. They're kind of there, but I would not write home about it as like a major thing. So yes, that is the data. It should be borne out, but it's not. So maybe we have to, maybe for purposes of invariance, it's easier to make bigger receptive fields. I'm not sure if that's a good question.